Well, I'd like to conclude this tutorial by talking about how memory changes across the lifespan. As I've already alluded to, we're familiar with the concept of learning to ride a bicycle in childhood and never forgetting. It does appear that as long as the motor system is competent to perform motor acts, there is a indelible lifelong storage of procedural memory that seems to be relatively robust against injury and disease that would afflict the nervous system. Sadly, that doesn't seem to be the case for our declarative memory system. We know that our memory system does indeed change across the lifespan with age and is particularly vulnerable to certain forms of neurodegenerative disease. Well, to just remind you of how the brain indeed does change across the lifespan, we achieve the peak size of our human brain probably at about the end of the second decade of life. Thereafter, for both men and women, there is a gradual decline in the overall size of the brain. So what's actually changing as we age through adulthood? Well, we know from studies in animal model systems that there are at least a few cellular components of the brain that are likely responsible for this slow, gradual decline in overall brain size. So what's actually responsible for this gradual decline in the size of our brains throughout our adult years? Well, based on non-human studies, we think that one cellular element that is responsible for this decline is a reduction in the numbers of synapses that are present in our networks. As you may recall from Unit 5 of the course, our synaptic density seems to peak in our adolescent years, and throughout the remainder of adolescent life into young adulthood, there is a rather dramatic decline in synaptic density on the order of 30 to 50% in many regions of the cerebral cortex. Thereafter, the decline in synapses is not nearly so steep, but it is an ongoing process. Now, in adolescent life, the pruning of synapses, the decline in overall synaptic density is part of normal, healthy development. It's a consolidation of networks that are responsible for who we are as unique human beings. However, throughout life, the loss of synaptic connections seems to gradually erode human performance in various dimensions of our lives, be it in motor performance or in cognitive performance, in one domain of cognition or another. Now, other changes that are taking place reflect changes in the structure of myelin around axons and white matter pathways. So while myelin is not necessarily being lost, although it may be in some regions of the cerebral white matter, myelin seems to be disrupted at an ultrastructural level. And as a consequence, the tight wrapping of myelin around axons in white matter seems to be loosened. And this in itself, we think, is an important consideration that explains functional decline across adulthood. One consequence of having this loosening, if you will, of myelination around axons is that the propagation of those signals is now subject to propagation failure, or at least to inefficiencies in the rates of transmission of information from one gray matter center to another. Now, it's also important to emphasize when we're considering normal, healthy aging that we are typically not losing neurons as our brain ages throughout adult life. This gradual decline that we see in brain size is due very likely to these kinds of ultrastructural features of networks and neuronal connections. But the actual loss of neurons, we now think, is pathological. That is to say, we normally are not losing neurons as we age. Our gray matter volumes are declining, but not because of the death of neurons. Now, sadly, there are individuals that are experiencing the death of neurons, but we consider that to be a pathological context. So cellular loss throughout the brain is not to be considered a part of normal aging. Now, I suspect that many of you may be supplementing your diet in various ways with products that are intended to increase cognitive capacity and memory function and other aspects of human brain performance throughout adulthood, or perhaps you're on particular dietary regimes that are designed to do the same. And you may be wondering, well, is there significant benefit from these kinds of dietary factors? I would suggest that we still don't understand well enough yet how these dietary factors impact brain structure and function. There may be benefit, but I think what is clear is that the benefit of aerobic fitness trumps the more modest benefits of any of these other factors that heretofore have been studied in terms of supporting brain function into adult life. So I think that's great news. I think what we need to appreciate is that sometimes the best solutions to our health problems are right in front of us and are often the most simple. And simply moving our bodies, increasing our aerobics fitness, seems to me anyway to be the most compelling single factor that will promote the health and the function of our brain throughout the lifespan. Now, quite sadly for many adults around the world, a serious concern with memory and aging is the development of dementia, and one particular leading cause of dementia, Alzheimer's disease. Now, we could easily spend an entire session in medical neuroscience together talking about Alzheimer's disease. Unfortunately, I've not planned to do so. For those of you who may have a special interest in this topic, I'll have to simply refer you to other sources that are available. Perhaps in the future we may develop a neuropathology course that will give us the time and the place to explore such diseases of the human nervous system in more depth. But for this session, I simply want to touch on this topic and perhaps inspire you to learn more on your own. Well, Alzheimer's disease is a neurodegenerative disorder that primarily afflicts the cerebral cortex. And here in this figure we see the distribution of pathological hallmarks of Alzheimer's disease, with these dots reflecting the density of those pathological changes. And what we can see is that the entire cerebral mantle can eventually be affected with Alzheimer's disease, but the disease process itself seems to be focused on the medial part of the temporal lobe and then extending into the parietal associational cortical regions. So on the lateral surface, there seems to be a broad region of the parietal temporal associational network that has severe affliction with the development and progression of Alzheimer's disease. So what these individual dots in this schematic actually represent are the two features that are hallmarks of this disorder. One is the presence of something called the senile plaque, which is an extracellular accumulation of protein and other kinds of cellular components. And in addition, the second hallmark is something that we call the neurofibrillary tangle. And this is an intracellular inclusion that is present within neurons that are very likely on their way to degeneration. And certainly when they present with a neurofibrillary tangle, that is an indication of dysfunction within the cytoskeleton of the neuron and the trafficking of important subcellular constituents that are important to support the function of the neuron. So senile plaques and neurofibrillary tangles are concentrated then in the associational regions of the medial temporal lobe and then the lateral temporal cortex into the parietal associational cortex. And the affliction of these associational regions helps us understand why in Alzheimer's disease one of the early indications of the onset of the disorder is a failure of our medial temporal lobe memory system. The distribution of these plaques and tangles also helps us understand why long-term memories are often preserved in these patients, at least prior to the terminal stages of the disease. It's quite common to find individuals afflicted with this disorder that retain memories from childhood, but have severe disability in their capacity to acquire and to consolidate new memory. And we think this clinical picture reflects rather well on the distribution of pathology within these associational regions of the cortex. Not always, but typically we find that association. Well, again, I feel like I need to apologize to those of you that may have been drawn to medical neuroscience because of a particular interest in Alzheimer's disease or other kinds of disorders and diseases of the nervous system. It's simply beyond the scope for us to talk in depth about the various neuropathologies that afflict the human brain. I hope rather I'm giving you a solid foundation that would support your explorations of these kinds of disorders, should that be where you want your studies of the brain to go in the future.